Assalamu alaikum, dear students. Today we will try to discuss the classification of bryophytes. The division Bryophyta has been divided into three different classes: liverworts, which are also known as Hepaticopsida, hornworts, Anthoceratopsida, and mosses Bryopsida. At this point of time, you cannot remember all the minute details about these three different classes. So, for the sake of convenience and considering the time constraints, we will try to remember some important distinguishing characters of each class. First of all, Hepaticopsida or liverworts. The word liverworts means the shape of the gametophyte in these plants is just like liver, which is lobed. That means their thallus is lobed. Okay? They are having lobes, and this thallus is dorsiventral and is appressed to the substratum. Dorsiventral means it is having two distinguishing surfaces. The upper surface is known as dorsal surface and the lower surface is known as ventral surface. You can see clearly in this image, this is the thallus of Mertensia, and it is having dorsal surface, which is upper surface, and the lower surface, which is attached to the substratum, that is known as ventral surface. Okay. And in some cases, in some Hepaticopsida, example Jungermania, the thallus is having leaf-like appendages, and these leaf-like appendages are arranged in two rows on the stem-like structures. Okay? Here this is Jungermania. You are having leaf-like appendages. Here you can see easily leaf-like appendages. These are arranged in two rows on stem-like structure. This is stem-like structure. Okay. The ventral surface of the thallus contains unicellular rhizoids and multicellular scales in case of these Hepaticopsida. Here you can see these are the unicellular rhizoids in case of Mertensia, which is a Hepaticopsida, okay, which belongs to Hepaticopsida. And in addition to these unicellular rhizoids, there are also present some multicellular scales. Okay. The function of rhizoids and scales is absorption of water and the scales help in preventing the desiccation of the thallus. जो scales होती है वो water को absorb करती है, and this helps in preventing the desiccation, drying of the thallus. Okay. Now the internal structure of the thallus, I mean the anatomy. In these Hepaticopsida, the thallus is divided into two regions. It has two distinctive regions. The lower region is known as storage region and the upper region is known as assimilatory region, okay, or photosynthetic region. The photosynthetic region contains photosynthetic chambers. Here you can see these chambers contain photosynthetic filaments, and these photosynthetic filaments help in photosynthesis. Okay, this is in case of Mertensia. Or in some cases, for example, in Riccia, the thallus, which is divided into two regions, the upper region is known as photosynthetic region and the lower region is known as storage region. Okay. The photosynthetic chambers are not present. That means you simply have photosynthetic filaments. You do not have photosynthetic chambers. Okay. You do not have photosynthetic chambers. You simply have photosynthetic filaments, and these photosynthetic filaments are closely placed to each other. Okay. Now the individual cells, for example, this is an individual cell of the thallus in case of Hepaticopsida. This individual cell contains many chloroplasts, and the chloroplasts do not contain pyrenoids, okay, because pyrenoids are present in case of Anthoceratopsida. This is a distinguishing feature. We can say that liverworts do not contain pyrenoids. Pyrenoids are protein storage bodies. These are lacking in Hepaticopsida, but they are present in Anthoceratopsida. Now their reproduction. In case of Hepaticopsida, reproduction takes place by vegetative and sexual means. The vegetative reproduction or asexual reproduction, it takes place by fragmentation. That means the thallus fragments, it forms different fragments, okay, and each fragment gives rise to new plant. Or it can also take place by gemma. Gemma are green multicellular structures, and these multicellular structures are actually asexual buds. These are multicellular structures which are produced in gemma cups. ठीक है बेटा, मैंने आपको पहले भी बोला था जब हमने जनरल कैरेक्टर्स पढ़े थे. ठीक है बेटा, ओके. Now the sex organs in case of Hepaticopsida are of two types. One type is known as embedded type. In case of embedded type, the sex organs are embedded in the gametophyte. That means if you have an antheridium, the antheridium does not contain stalk. ठीक है? The antheridium and orchigonium, they lack stalks, or they can be of projected type. For example, Markensia. In Markensia, you have an antheridium, and that antheridium contains a well-developed stalk. You have an orchigonium, and that orchigonium also contains a stalk-like structure. Okay? Or simply you can remember that in case of projected sex organs, the sex organs contain stalks, while as in case of embedded sex organs, the sex organs does not contain stalks. Now they are sporophyte. The sporophyte in case of these liverworts is either constituted of foot, seta and capsule. Example is Markensia. कुछ liverworts में जो sporophyte होता है, that is divided into foot, seta and capsule. And in some cases, for example Rixia, it is only constituted of capsule. There is no foot and seta. And one more thing that you have to remember is that in these sporophytes, in the sporophytes of liverworts, the columella is absent. Columella means central sterile tissue. ठीक है? जब हम एंथोसेराटॉप्सिडा पढ़ेंगे तब आपको समझ आएगा columella क्या होता है. ठीक है बेटा? And after meiosis in the capsule, ठीक है? After meiosis in the capsule, the spore mother cells, they form spores, and these spores after germination they give rise to gametophyte directly. ठीक है? They form gametophytes. Okay beta? This is